Good morning, everyone, and welcome to Genome Webinars. I'm Ben Butkus, Editorial Director for Genome Web, and I'll be your moderator today. Today's webinar is entitled Mining the Post-Mortem Human Brain for Neurodegenerative Markers, Deep Sequencing of Cell Type Specific Transcriptomes. The sponsor of this webinar is Neugen. Our presenters today are Steve Sheardown, Head of Molecular Biology and Cell Engineering at Cerevance, and Ashesh Saraya, Product Manager at Neugen. A reminder to attendees, you may type in a question at any time during the webinar. You can do this through the control panel, which usually appears on the right side of your screen. Click on the Q&A box in the upper right side of the control panel. When you click on Send To, please select All Panelists. We will ask the panelists questions after their presentations. We'll now begin with Steve Sheardown of Cerevance. Please go ahead, Steve.
Well, thank you, Ben, and good morning or afternoon or evening to all participants, whatever time zone you happen to be in. It's a slightly cloudy afternoon here in Cambridge. I'd like to thank the organisers for inviting me to present the Cerevance transcriptome Cerevance transcriptomic technology and pipeline today. So I will begin with an introduction to ourselves, Cerevance, and who we are and what we do and why we exist. An introduction to NETSseq, which is our drug target discovery platform, a transcriptomic platform. I'm going to talk for about half of this presentation about how we, in molecular biology, took NETSseq from proof of concept, small scale, to a high throughput pipeline process and the challenges that we met along the way and solutions that we've arrived at. And also, and at the end of that part of the presentation, I'll present some examples from our own NETSseq data that just demonstrate that the pipeline's really working very well. Okay, so Cerevance is a young company, about a year and a half old now, headquartered in Boston, US, with our research facility in Cambridge, UK. We were conceived to make use of NETSseq, which is a powerful sequencing platform invented by Xiao Zhu and Professor Nat Heinz at the Rockefeller Institute in New York. So the NETSseq method enables detailed transcriptomic profiling of defined cell populations within the human brain. And our goal is to use this platform to generate novel information and insights and ultimately drug targets to address neurodegenerative conditions where there's still substantial unmet medical need, including but not limited to Parkinson's, Alzheimer's, ataxia, and Huntington's. And here's the problem, really. The brain contains hundreds of diverse cell types. It's an enormously complex organ, as everybody knows, and neurodegenerative conditions typically translate quite poorly from animal models. So while engineered lineage markers in rodents have been very informative in many respects, they do have limitations, such as species-specific differences in cell numbers and proportions, and also, to an extent, expression patterns within cell types, and we've got a little data to show that later on. So to address human disease, we strongly feel that we need to be working with human systems, but how to do that from native tissue? So a large body of cell type specific gene expression is readily available in the literature and via online resources like the Allen Brain Atlas. Using this kind of information, we can fairly easily identify markers that distinguish cell types of interest to us, and as this figure shows. So here we have brain sections showing cell type specific nuclear labeling, which is what we want because it isn't possible to isolate intact neuronal cells in useful numbers from frozen post-mortem brain due to their size and fragility. But we can isolate nuclei because these are much less susceptible to freeze-thaw damage. So nuclear markers are what we're interested in. And if you do that, and you label nuclei with cell type specific antibodies, what you can then do, of course, is fluorescently label them and subsequently FACS sort into pure populations, which you then sequence. Now, one thing that I should mention at this point is that in order to do this, one of the parts of the method of the procedure is fixation, formaldehyde fixation. You can't easily sort unfixed nuclei because they're just a bit too fragile to get through the process. So we have to fix them, and that unfortunately reduces RNA quality, which I'll show you very shortly. Now, moving on to the Cerevance NETSseq workflow. So this is the workflow that we developed essentially over the first half of the existence of our company. So starting from a disease-relevant cell type, we ID unique, hopefully nuclear markers, plus a panel of antibodies that correspond to those markers, and these then pass into a histology, IHC, and also a FACS evaluation pipeline. Anything that gets through that pipeline then goes into a full-on sort, and that means sorting across from a panel of control and donor tissues, a variety of different brain tissues usually, and the output from that, the nuclei come out. The nuclei get passed on to my team, my molecular biology team, and we extract RNA, convert to sequencing library that then ultimately ends up as data back in our bioinformatics and target discovery pipeline. So that's how the whole process works. Now, the whole left-hand side of this slide, the marker selection and the histology and the FACS, etc., that actually represents a very large amount of work for a well-coordinated team. And so I'm doing a little bit of an injustice here, I feel, because I'm not actually going to talk about any of that. I'm just talking about the bit after the FACS, the molecular biology, and a little bit of the transcriptomics. So our challenge was to make the molecular biology, was to move the molecular biology from a low throughput proof of concept platform into a high throughput process. And what are the challenges involved in that? Well, I've already said that we have to formaldehyde fix the nuclei. The tissues are post-mortem, so they're scarce and they're valuable, and also they're somewhat variable, and I'll show you that in a moment. There's a little bit of degradation during processing. We're working with nuclear RNA, which is not as easy to work with as whole cell RNA because the DNA-RNA ratio is higher, and that has a carryover contamination aspect. And we have to process up to 96 per week, and so whatever we do, whatever process we design, it's got to be plate-based. So what we needed was a very broad operating window for whatever amplification system we chose, a very efficient RNA extraction process, very efficient gDNA removal, and whatever we do, it needs to be automation friendly. So just to show you what fixed RNA looks like, because a lot of people don't know this. So what we have in this panel is, well, are two bioanalyzer traces. The one on the left is from unfixed nuclei, and that's very, very similar to total cell RNA. You can't really tell at a glance. Now, the ribosomal peaks are sometimes a little bit different in ratio compared to total RNA, but essentially it's the same. And it's not unusual to get a RIN number that's in the kind of 7s, 8s for that reason, because the algorithm depends on ribosome to total RNA ratio. The panel on the right has been fixed. It's the same nuclei, fixed and extracted with an FFPE extraction kit, RNA extraction kit. And what's happened is that the RNA is fragmented substantially. So this is a question that people ask me all the time, and it drives me mad. You can't use RIN number to express how intact fixed RNA is, or fixed and extracted RNA is, because it's fragmented, and so the algorithm intrinsically doesn't work. But what Illumina suggests is this figure DV200, which is simply a measure of how many fragments are over 200 nucleotides in length, which is quite tractable for a library. And so it's a pragmatic measure and a perfectly reasonable one, but you can't refer to the RIN number because it does not work. Now, that RNA that's on this, that's on this slide, this is really high quality stuff. This was fresh nuclei, freshly isolated. It's as good as it gets. You never actually see it better than this in my experience. Because what we're working with is post-mortem tissue. And this is a representative panel from a set of the post-mortem tissues that we received. I've run a lot of these panels through the bioanalyzer. They all pretty much come out with these kinds of profiles. It's variable, to say the least. Some of these are great, others are not so good, some are dreadful. Problem is, of course, this is very valuable tissue, and it's not very easy to go back and ask for more of some of this stuff. So really, we need to be able to use all of it if we can. And we have no control over how it's treated before it arrives in our facility. So obviously, we need reagent systems that can handle most of this variability. The other thing is that when you process tissues, or process nuclei rather, well, invariably there's a little bit of degradation as you go through staining and sorting. You can't avoid it. You do our best to avoid it, but you can't avoid it entirely. So what we get in molecular biology is a wide range of input material, and so we absolutely have to maximize RNA recovery and amplification. So what we did, the first thing that Jason Lawrence is a senior molecular biologist in the team, and we did this together earlier last year. The first thing that we did was to look at RNA extraction and library amplification kits, and to carry out a fairly extensive comparison to find out which of these, which kits actually fit the bill and which ticked the most boxes. And we went in with an open mind. I mean, neither of us had actually worked with FFP RNA before, for instance, so we had no favourite kits in mind. We just tried everything that we could think of, as far as the RNA was concerned, at least. So the RNA extraction was pretty easy to work to optimize, though. And so we compared 5 different FFPE RNA extraction kits, and we used bioanalyzer traces, and we used qPCR, and we also put some of them through full sequencing. And essentially, quality-wise, all of them were great, really very good indeed. But the Norgen spin column kit, based on yield, really, just won. It was a little marginal, but the Norgen kit was marginally but consistently better than the rest. Unfortunately, it was the columns that worked best and not the plate-based kit, and so we had to get a little bit creative, and we bought a 3D printer and printed SBS footprint manifolds that enable us to process spin columns in 4x24 column format. That's multi-channel compatible, so we can process anything up to 96 samples in parallel in a little under 3 hours. So that works really well, and it's really versatile. So if you're thinking of setting up automation in your lab, buy a 3D printer. Really, you'll thank me. Which brings me to the RNA-Seq amplification kits comparison. In this case, we actually used 2 kits that we were already familiar with, and very happy with both, I must say. And what we were looking at here was yield and reproducibility, of course, but significantly deep sequence from very low nanogram sample input, with minimal artifacts, obviously, and ease of automation, because this was going to have to be automated. And we went into this quite deeply, and we made 54 direct comparisons all the way to deep sequence, including biological and technical replicates. And here's a little snapshot from one of those comparisons. This was a technical replicates comparison, where we divided a single RNA sample 8 ways and ran 4 replicates each using the Neugen and Sigma systems. And the first thing to note is that consistency within kits was uniformly excellent. It was really very, very impressive. And both amplified well from low nanogram inputs as well, I must say. But if you focus on the red boxes, what we did notice was that there are differences between the kits. There are biases between them. And one kit will give you higher read counts for one gene, the other kit will give you higher read counts for another gene. And that's quite common. The example at the top is a bit extreme, and I picked it out just to make the point, but you can scroll down the read counts, and this is gene counts in TPM, you can scroll down the read counts and you can find a lot of these. So what was apparent was that we can't cross-compare between different kits. So once we'd made a selection, we basically, that was a committed step for us. So we made a big effort to make the right choice. And what we ended up with was we selected the trio kit for the following reasons, because it basically ticked all of our boxes. So it does cope with a very broad range of input RNA quantity and quality. Isothermal amplification is great. It goes to completion without optimization, and that's important if you're working with plates, because you can't treat samples separately. You just have to treat them all the same, and it has to work. gDNA carryover is low, and there's an initial DNA step integral to the protocol, and that's important when you're working with nuclear RNA. Starting volumes better than 10 microliters, also important when the RNA concentration is low, so you can maximize the amount that you add, and it really helps with liquid handling apparatus. You don't want to be pipetting small numbers of microliters. It's not that consistent across the plate, despite what manufacturers tell you. No need to sonicate was a big one, which was an issue with the original Ovation system, and we didn't really fancy sonicating 96 samples per week, so it was nice, very nice indeed, when the trio came along with no requirement to sonicate. And to be fair to Sigma, you don't need to sonicate those either. But really, the NuGen trio has been designed with automation in mind. So liquid handling volumes are compatible with robots, and it's a very thoughtfully designed protocol that really minimizes plate changes and pipetting steps and keeps those liquid volumes up. And Jason worked with with NuGen to optimize reagent volumes for the 96-well trio automation kit, so that they would work on our or to bring them to a point where they work extremely well on our Agilent system. So we kind of beta tested the kit, if you like. So those are our reasons for choosing the NuGen system. Now, I just want to take a moment to consider sample management, which is an incredibly important aspect of a high-throughput setup. So in our case, going from nuclei to library involves a lot of vessel and format changes. So we go from tubes to columns to several plate swaps and back to tubes. And really, the scope for human error during a process like this is pretty high, and we felt that it was important to put some effort into mitigating the problem. So from very early on, we decided that we had to move this whole workflow onto automated platforms exclusively. And Jason, who is not only our senior molecular biologist, but also automation expert, worked with Joe Donheath from NuGen to input the initial Agilent scripts and has worked on a lot of optimization of those scripts since then, and also configured our Cryogen biorobot to normalize and pull libraries at the end of the method. But what Jason also built in was barcode-based sample tracking. So our process actually looks like this. So every sample from the outset is barcoded, initially 2D barcoded lids, but then subsequently as well locations on barcoded plates. All of our devices are fitted with readers, and at each vessel change, tubes or plates are rescanned and remapped to the appropriate container. And the software even generates our internal and external sequence submission forms. And a sequencing supplier, by the way, seeing as I'm naming names, is Source Bioscience in Nottingham. And we feel that removing the human factor from sample tracking provides a level of data integrity that's critical to any kind of high-throughput pipeline. If there were a transposition error, we probably wouldn't spot it, and it would really destroy an entire data set, so you can't afford to risk it. Now, moving on to some more RNA. So how well does the trio kit perform in real life? So these are real samples that have gone through our pipeline. Our RNA is normally better than this, especially better than the two bioanalyzer trace samples at the bottom, but which look terrible. But nevertheless, isothermal amplification is incredibly robust, and even with this degraded material, we got everything. All of these got through sequence QC. I'll talk about QC shortly. This is obviously critical for us because it means that we don't waste samples, that we get valuable information from almost every sample that we put through, despite the fact that some of them end up looking like that. And in fact, we don't even QC the RNA anymore, because really, what's the point? I mean, what would you conclude from these traces? You'd throw several of these away, and yet they came through with good sequence. So a typical weekly process looks like this. It's basically a 3-day process from RNA to library, and on the 4th day we send off our pooled libraries to the sequencing lab. I guess the one point that I want to make about this particular slide is that, again, from the outset we separated our pre-amp from our post-amp labs, and physically separate labs, not just separate parts of the same lab. And this is something that I've always had a bee in my bonnet about. If you're running a lot of samples through a fairly large scale amplification, the chances of getting contamination are quite high. And if you do get a contamination, you wouldn't know for 4 or 6 weeks, and you'd have a hell of a problem fixing it. So we can't afford to make that mistake. And so from the outset, we separated these processes. And over the past 8 months, we've been operating this pipeline routinely now, and it's working very well indeed. And we've added over 1,500 transcriptomes to our database, and we expect to double that by the end of this year. Okay, so that's the pipeline, and that's how Jason and I configured it. And now we'll come on to some data, because I've spent a lot of time describing this, but the real question is, does it actually deliver? So first, a few details about sequencing in QC. So the platform that we use is the Illumina high-seq 75 base paired end reads, and at 32 samples per pool, on this particular platform, we're averaging 25 million reads per sample. We use 2 QC methods. We use TIN, or transcript integrity, which is a score that provides a metric representing total transcript coverage compared to the whole transcriptome. I've referenced the original paper below if you wish to get some more information about that. And we also use percentage intergenic reads as a measure of genomic contamination. Our cutoff values are 14 and 20% respectively, which our bioinformaticians tell me are quite stringent. I'm prepared to take the word for that. I'm not a bioinformatician, but actually we can usually derive useful information from samples that fail to meet these criteria, and the kind of thing that you would still be able to discover, for example, is confirming cell type ID, measuring high to medium expression changes, but those samples wouldn't form part of our core data set. Nevertheless, better than 90% of the material that we've put through over the past 8 months has passed QC, and so we feel that this is a very good success rate from post-mortem tissue. The other thing I want to say is that, okay, I've already said we've added over 1,500 transcriptomes so far. Mostly these have been control and Parkinson's. We are expanding into other areas such as HD, ALS, AD, and others. These are samples that are kind of queued for processing. We've built a relationship with several brain banks worldwide to access control and disease tissue, and at the moment we have a little over 3,000 clinical samples in our freezers, but this is expanding all the time. The strategy that we're using is to produce transcriptomic data from 20 individuals per cell type and brain region over an equal split of males and females within defined decades. And thus far, we've validated 24 unique cell types, these are cell types that we know that we can sort from a variety of brain regions reproducibly, and we have many more in validation QC. And our first NETSeq-derived target has entered early phase drug discovery. So that's all going rather well. Let's look at some data. So here we have an example of a panel of sorts from 16 individuals with cell type populations gated in red. One thing that's immediately noticeable, well, it's noticeable if you look hard at the figures at least, is that there's a bit of variation between individuals. These populations appear in slightly different places on the FACS, and this is common actually. We see this a lot, but it doesn't affect our ability to gate off pure cell populations. And so and the right-hand panel shows some shows some expression markers. This is from the IGV viewer. And the cell types that we want can all be reproducibly sorted to a high degree of purity, and the markers that we expect to see, we do see, and just as importantly, those that we don't expect to see are largely absent. This is a slightly more detailed figure, or slightly larger data set. So in this example, we have 12 cell types mapped against known marker genes, which we expect to be there, and expression level, as it always is in these heat maps, is indicated by the color code, the orange-red being the highest. And once again, the correspondence between expected and observed markers is very high, and so we are confident about the purity of our cell types. Now, this next heat map is my favorite image. Actually, it's a beautiful image. This was constructed from our recent transcriptomic data using a clustering algorithm that highlights similarities. So we're not looking for known marker genes here, we're just looking for similarities. Again, high expression is towards the red end of the spectrum. And these represent sorts of 4 cell types from 3 brain regions from a panel of donors, and this is indicated on the 3 top bars on the x-axis, with genes and expression levels plotted on the y-axis. And again, we consistently see clear and reproducible expression patterns that correspond very tightly to pure cell populations. But if you zoom in, what you can also see is that you can find reproducible expression differences between identical cell types isolated from different brain regions within the same donor. Given the differences in cellular environment, I don't actually think that's very surprising. And of course, there are a lot more similarities than differences, but it will be interesting to map out these region-specific differences and look for correlations with function in different brain regions, and to ask whether any disease states have a region-specific differential effect, and it won't surprise me if it does. But NETSseq offers a very powerful way to do this. And while I'm on the topic of expression, I said way back that there were expression differences between rodent and human. This is a very nice panel that Xiao put together, showing really the most blatant expression differences for given cell types from the same brain region between mouse and human. And really, of course, there are more similarities if you look at the whole data set, but nevertheless, these are pretty plain differences, and there are others that aren't shown. So I think it just makes it all the more obvious that really, if you're asking questions about human problems, you need to be looking at human material. Now, the last data slide, really, and this is a question that I'm sure that somebody's getting ready to ask. We've certainly been asked it before, and the question is, why are we going to all this trouble to pull cell nuclei in light of the recent single cell and nucleus RNA-Seq publications? And that's a perfectly fair question. Well, mostly it's about depth of coverage, and that may change as single cell technology matures, but right now, as the charts illustrate, the number of genes that return 0 counts from single nucleus sequencing is much higher than the same count for NETSeq. It's almost 20,000 compared to about 8. And so single nucleus sequencing currently misses a lot of genes that are expressed at a low level. And on the same basis, you would also expect to miss subtle expression changes for genes that you can see, whereas with NETSeq we won't miss these. Now, I'm not saying that we won't use single nucleus sequencing for some purposes. In fact, we're setting up right now with a view to novel cell type identification, or where target genes are known and highly expressed, or even as a means of accessing a cell type from an enriched population where there's no good unique marker. However, NETSeq has clear advantages in situations where we wish to sequence relatively rare and interesting cell types like human Purkinje cells, or in situations where a disease state may have depleted the population. And a good example of that is loss of fast spiking interneurons in Huntington's. It's difficult to see how you would get enough sequence for them by single nucleus sequencing at the moment. And so in cases like these, the ability to purify and enrich populations from a heterogeneous mix is a really big advantage that NETSeq has to offer. Okay, so I'm pretty much at the end of my presentation now. I seem to have rattled through that a little more quickly than I expected to. Never mind, more time for questions. So really, to summarize, the RNA-Seq library assembly process, well, By combining the best available sequencing technologies that we could find with automation, we've at Cerevance have established NETSseq as a robust and powerful high-throughput drug target discovery platform, and this really works. And the NuGen Trio kit, with its impressive ability to convert suboptimal RNA into tractable library material and an automation-ready workflow, has become a key component of our molecular biology pipeline. And we've succeeded in our goal to establish a high-throughput pipeline to mine deep transcriptomic profiles from defined CNS cell types, and we've produced over 1,500 high-quality transcriptomes to date, with many more to follow. We're producing an extensive database with unique cell type-specific gene expression profiles for neurodegenerative disorders, and we firmly believe that this will lead to novel insights into brain cell function and dysfunction in the disease process. Ultimately, of course, the goal is to identify novel drug targets for CNS diseases with high unmet need, and we believe that we're making excellent progress toward realizing that goal. So thank you very much for calling in and listening, and now I will hand back to Ben.
Thank you, Steve. A reminder to attendees, you may type in a question at any time during the webinar. You can do this through the control panel, which usually appears on the right side of your screen. We'll now continue the webinar with Ashesh Saraya of NuGen. Please go ahead, Ashesh.
All right. Thanks for the introduction, Ben, and for a great webinar, Steve. So NuGen has a broad portfolio of innovative kits that address a wide range of sequencing applications. You just heard about the Trio kit, but we also provide kits for DNA-Seq, methyl-Seq, and SNP genotyping applications. So focusing on RNA-Seq, NuGen is a known leader in providing RNA-Seq solutions for low-quality, low-input samples. The same technology, however, can be applied for higher input samples or higher quality samples, providing streamlined and robust solutions for any input level or sample type. All of our complete RNA-Seq kits also integrate transcript depletion using our NE-deplete technology to remove unwanted reads from the library. So just a little bit more detail about the Trio kit. The Trio kit provides a robust solution for low input and degraded RNA samples by combining 3 of NuGen's core technologies. The single primer isothermal amplification, or SPIA, is a highly validated method that converts RNA to amplified double-stranded cDNA. This upfront amplification increases the number of copies for each transcript, increasing the chances that low or rare expressed transcripts are converted into a library and can be therefore detected during sequencing. The second technology, dimer-free adapter ligation, eliminates the formation of adapter dimers, enabling efficient adapter ligation regardless of inputs and without the need for adapter dilution or titration. Finally, the NE-deplete technology efficiently eliminates unwanted reads from the library, increasing the number of informative reads or enabling higher multiplexing for more cost-effective sequencing. Unlike Ribo0, this depletion actually occurs after library prep, enabling us to do transcript depletion regardless of input and eliminates upfront manipulation of precious RNA samples. This enables Trio to overcome multiple NGS challenges. As Steve mentioned, sample inputs are rarely uniform in quality or quantity, especially when starting with precious samples. The SPIA technology efficiently amplifies all samples regardless of input quality or quantity, reducing the effective sample variability, and it eliminates the need for downstream protocol optimization. The robust Trio workflow ensures results for your precious samples, reducing the time wasted due to library prep failure or having to re-prep samples. And secondly, the SPIA amplification also increases the copy number of each transcript in the sample, increasing the dynamic range and enabling better detection of rare or low copy transcripts or pathogens. Finally, the integrated NETSeq technology eliminates ribosomal RNA reads from your libraries, so you get more informative reads per sequencing run. NETSeq is also customizable so that other highly abundant transcripts, such as globin, mitochondrial transcripts, or other high abundant transcripts can be removed, reducing your sequencing costs. Steve provided a great overview of TRIO's advantages in neurobiology. Another application where TRIO provided multiple benefits is viral detection. We collaborated with Dr. Daryl Dinwiddie at the University of New Mexico to test the ability of TRIO to detect respiratory viruses in nasopharyngeal swab samples collected from patients with respiratory difficulties. Nasopharyngeal swabs provide a minimally invasive and low-cost method for sample collection. However, these samples are challenging because of low RNA amounts, poor RNA quality, and significant host RNA contamination. The ability of TRIO to detect these respiratory viruses in these samples was compared to a hybrid capture target enrichment method of known or expected viral genomes. Well, we assayed 12 samples with varying inputs from non-detectable to about 100 nanograms per microliter, as shown on the left side of this table. Samples shown in green indicate that a respiratory virus was detected either by the reference method or by TRIO. Of the 12 samples that we assayed, the reference method was able to detect a respiratory virus in 6 of the samples. The failure of the hybrid capture method to detect the viral pathogen could be due to RNA degradation, which reduces the efficiency of conversion to a library, or reduce the ability of the hybrid capture method to efficiently capture the viral sequence. Alternatively, TRIO was able to detect a respiratory viral pathogen in 10 of the 12 samples, demonstrating that TRIO can efficiently detect respiratory viruses from these sample types. Additionally, TRIO is a whole transcriptome approach, which enables it to detect viral fragments that the hybrid capture method may fail to capture. Interestingly, respiratory viral pathogens were not detected in 2 of the samples by either method. Since TRIO is a whole transcriptome unbiased method, sequencing data generated from the TRIO libraries could also be used to detect other potential pathogens. In this sample where a respiratory virus was not detected, human RNA and unidentified sequences represent about 60% of the reads. However, the data also showed the presence of coronavirus, hep C virus, as well as a number of other opportunistic bacterial pathogens that could provide more insight into the patient's symptoms. So, as we've heard from Steve, and as I've shown, TRIO provides a unique solution for whole transcriptome RNA-Seq from inputs with variable quality and RNA quantity. The integrated SPIA and NETSeq technologies enable robust detection of rare or low-abundant transcripts and pathogens from a broad range of sample types. Thank you, and we're happy to answer any questions.
Thank you, Ashesh. Before we enter the Q&A portion of the webinar, we would like to ask attendees to please take a moment to take our exit survey after the webinar has ended to give us your feedback. We'll now enter the Q&A portion of the webinar. First question is for Steve. Will the data sets that you generate be freely available or only through collaboration?
Well, at the moment, they're proprietary, and so no, that they won't be freely available in the immediate future. They may be available, well, they're available through collaboration, yes, but who knows what happens a few years down the line, but at the moment, no.
Thank you. The next question is also for Steve. Can you highlight the advantages of your approach versus array-based expression profiling typically done in brain bank data?
Well, it's a much more quantitative approach, really. That's array-based expression profiling is not as neither as deep nor as accurate as RNA-Seq, in my view.
Thank you. The next question. There is a move towards cytoplasmic RNA sampling rather than using total RNA in order to improve correlations with protein expression. Have you completed any correlations with protein changes?
Okay, we haven't carried out any correlation with protein changes. We couldn't do it from this material, actually. But Xiao at Rockefeller, when she was at the Rockefeller, did carry out a comparison between nuclear RNA and translated RNA, so by pulling down polysomes from cells in which neuronal cells in which she also isolated nuclear RNA. So we were not comparing protein with RNA, but with comparing translating RNAs with nuclear RNA, and the correlation between those was actually very good. But no, we haven't looked at any proteomics. It'd be nice to, but I don't really see how we would do it with this system.
Thank you. The next question. You discussed analysis methodology, but this is all ex vivo data. More recently, there is ongoing discussion regarding post-mortem transcriptome limitations. Can you comment on that?
I can't comment on that because it's not something that we've had any opportunity to compare. I mean, are we talking about completely fresh material isolated from a brain as compared to material that's been, say, in a chiller for 24 hours? I mean, is that the question? I'm not really very sure what the question is. What I can tell you is that we have compared transcriptome data from post-mortem material that's been maintained chilled for up to 80 hours, for anything from 24 to 80 hours, and we don't actually see any differences. So the RNA integrity seems to be really very good across quite a large time span before the brain's frozen.
Thank you. Yes, the participant clarified that the question was in vivo versus ex vivo.
So you can do that experiment with rodents, and Xiao has done those experiments. I don't have the data in front of me, but the post-mortem material compares very well with fresh material.
Thank you.
And we actually did a small study where we did something similar with rat, actually, and we aged out rat brain just by dissecting and keeping it in the fridge for up to 4 days, and we compared transcriptomes from the beginning with fresh brain and from the end of that process, and again we didn't see any real differences. I don't have that data in front of me either, but we did do that experiment.
Thank you. The next question is for Ashesh from Nugen. What other depletion probe sets are available for the TRIO kit?
Sure. So we have obviously human. We also have a probe set available for mouse with globin depletion as well as ribosomal RNA. We have a custom probe set that is for pig ribosomal RNA, but we don't typically generate lots of different organism probe sets, but if a customer does need it, we design them specifically for that customer.
Thank you. The next question for Steve. Do you think the RNA would be different if the brain were fixed in PFA?
I doubt it. I don't think it would. It may be that well, the extraction process might be a little bit different. But really, it's a question of how fragmented the RNA is and whether you can sequence that fragmented material. And it's not massively fragmented by, or it is quite fragmented by formaldehyde, but it's more about the age and treatment of the samples before they get to us. Really, I doubt that it would be different, but I don't know because we haven't tried.
Thank you. The next question is for Ashesh. What is the minimum RNA quality that can be used as input into the TRIO kit?
Yeah, that's kind of a complicated question. As Steve mentioned, we would recommend using the DV200 metric that Illumina has proposed. For samples with DV200s of over 40 or 50%, we feel very confident that the TRIO will work. With RNA that's of lower quality, the TRIO will also still work and produce libraries. It's just going to be a question of the quality of the data. So as the RNA becomes more and more fragmented, the overall quality of the data is reduced. But we feel that for most degraded material, the TRIO kit works very well.
That's the reason why we use 75 base paired end sequencing, because really this material is very degraded, and using a longer read than that really wouldn't gain us anything. And we did start measuring DV200, but honestly, I didn't see much of a correlation between DV200 and the quality of the sequence that we got out at the other end. And one of the problems with using that as a measure is if you have very low yields, you start to run into electronic noise, and then the bioanalyzer gives you back a number that isn't really real, so you can't always really trust it exactly. And so if you have both low input and low quality, you can't even measure it in that way, and you just basically have to put it in and see if the outputs meet your QC criteria.
Thank you. The next question is for Steve. Have you had an opportunity to compare samples between different stages of disease advancement?
Yes, to a small extent with the Parkinson's material, but we haven't got very much disease data yet. So we've asked for different disease stages from various different brain banks, and we have them. It's actually quite difficult to get enough material of different defined disease stages, but we can access it. But yeah, we've got a lot of samples queued. At the moment, really, we're concentrating on using a single disease stage because that gives us a reasonable number of data sets that we can compare. But the answer is yes, but it's on the way. We know that it's important.
Thank you. One moment, please. Next question is for Steve. What is your sample throughput in terms of cells per week that you can process? Is this easily scalable? Okay,
well, molecular biology is scalable because we have plates and robots. So well, I have Jason sitting in the room with me, and he runs the RNA and library prep processes. We can run 96 comfortably. I think we could run twice that. And he's nodding. So yeah, we could. The difficulty actually, and the part of the process that's much more linear, is the sorting, because the fax is more of a bottleneck. So if you want to double your output, you need to double the number of machines and the number of people to run the machines. So really, scaling in molecular biology is fairly easy. Scaling with the sorting is less easy. But 96 per week is certainly achievable with the setup that we have now. I mean, if we needed to double it, we could, but it would be expensive.
Thank you. Next question. For the post-mortem brain tissue, what is the usual time between death and freezing, and does that have an effect on RNA quality?
Okay, well, that's a good question. It's generally below 24 hours, but we have run sequence from samples up to about 80 hours, and we've not really found any difference in RNA quality, which I found quite surprising, I must say, but really there isn't. I think probably more important is the time between death and chilling, rather than 4 degrees and freezing. I suspect, but I don't know.
Thank you. Next question, how many nuclei do you typically use per sample?
That is highly variable. It depends very much upon the proportion of upon the frequency of the cell type within the sample. Ideally, we'd use about 30,000 or so. The yields from this material are much lower than you would get from fresh nuclei, so we want to start with as many as we can. But about 30,000 is a good number. 10,000 is also a good number. We have produced sequence from as little as 200 or 300, but you're really pushing the envelope at that point because it is degraded material. So generally 10,000 upwards is optimal, we find.
Thank you. One moment, please. The next question for Steve. Have you looked at pH of the brain tissue versus RNA quality, as this was thought to correlate better than PMI?
Yes, that is a very good point, and we haven't, but it has been raised, and it is something that we're going to do. But we are aware of that, and so yes, we will, but we haven't.
Thank you. One moment, please. Next question is for Steve. If you were designing a case/control experiment, for instance, human post-mortem controls versus Parkinson's disease, can you speak to the design considerations of using a drop-seq-based method versus a lower cell throughput, higher depth method?
Oh, that's not something I've really thought about very much. You would. I don't suppose you'd use a different amount of starting material. Well. No, you've put me on the spot there. I'd have to think that through. Sorry.
Okay. As a reminder to attendees, you can ask a question at any time in the chat box on the right side of the screen. If we don't get to your questions, attendees will be able to answer them following the webinar. The next question is for Steve. Can you clarify a little bit more about what 96 per week means? Does this mean one cell type for one brain?
No, 96 per week means 96 samples, and that could be multiple cell types from multiple tissues and multiple brains. It refers to the number of samples of nuclei that come to us. And what we try to do is we try to get as much out of a sample as we possibly can. And so you may sort for 3 cell types simultaneously, and those would all come out of the fax from one piece of tissue. So no, it's not one cell type for one brain. It's multiple cell types for multiple brains.
Thank you. That concludes the Q&A portion of the webinar. We'd like to thank our sponsor, Neugen, and our presenters, Steve Sheardown of Cerevance and Ashesh Saraya of Neugen. If we didn't have time to get to your questions, we will try to have our panelists answer them directly afterward. As a reminder, please look out for the pop-up survey from the sponsor after you log out. If you missed any part of this webinar or wish to listen to it again, A link to an archived version will be emailed to attendees. Thank you for joining us for this genome webinar.